Hi, my name is Gonzalez and today I'm gonna talk about the effect of thermal and high pressure treatments on the antiretroviral activity of human milk fractions. Rotavirus. It's a genus of double-stranded RNA viruses. There are 9 species of rotavirus referred to as a group A to I. Humans are primarily infected by species A, B, and C, most commonly by species A. The genome of rotavirus consists of 11 unique double-helix molecules of RNA. Each helix is a gene. Each gene codes for one protein except gene 9, which codes for 2. These viruses cause gastroenteritis. The symptoms include diarrhea, vomit, fever, and dehydration. Antiretrovirals. Rotavirus vaccines. It's an inactivated virus. This allows the infant to obtain antibodies against it. Human milk. It provides essential nutrients and protects the baby against pathogens. One of the mechanisms it has is associated with glycoconjugates whose glycan moieties may act as decoy receptors, hampering pathogens binding to the cell surface. There are few studies regarding the antiretroviral activity of human proteins, especially immunoglobulin A and MFGM. Aim. The aim of the present study was to evaluate the rotavirus neutralizing activity of different human milk fractions and proteins such as immunoglobulin A, LF, LIS, alpha LA, MUC, LDH, and caseins, and compare the effect of several heat and pressure treatments on antiretroviral activity. Methods: cell culture and rotavirus propagation. The rhesus monkey kidney cell line MA104 was used to propagate WC3 bovine rotavirus strain. Cells were cultured in MEM. Obtaining human milk fractions. Mixed samples were obtained from 10 donors. Milk was centrifugated, obtaining skim milk, and they added potassium oxalate to it. By centrifugation was obtained casein, cream, MFGM. Human whey was filtered through glass wool. Purification of human proteins. Immunoglobulin A were isolated by size exclusion chromatography. Human LF and LIS was purified using cation exchange chromatography. Alpha LA whey was obtained from skimmed milk and subjected to health filtration chromatography. MGFM, MUC, degradation incubated ultrafiltration. LDH was obtained by resuspension of MFGM. Recombinant human LF were obtained from rice and Aspergillus awamori, rice, holoform, and as isolated. These proteins were analyzed by SDA's page. Recombinant human LIS was obtained from rice. Its purity was determined with SDA's page. Rotavirus infectivity assay. Human milk fractions and proteins were mixed with trypsin-activated rotavirus suspension and incubated to evaluate the neutralization activity. Serum-free MEM and rotavirus suspension were included as negative and positive control respectively. Virus-infected cells were detected afterwards by immunofluorescence. Treatment of human fractions. The protein concentration of the sample selected for thermal and HHP treatments were those showing neutralization activity above 80%. The samples were placed in glass vials and heated in a thermostatic bath. HHP. Aliquots of pulling human whey were introduced into polyethylene bags, heat sealed and pressurized. They were put in a chamber and pressure was increased. SDA's page used to separate macromolecules. A polypeptide chain binds a amount of SDS in proportion to its relative molecular mass. The negative charge on SDS are strongly attracted towards an anode in the electric field. Gel restrain larger molecules from regating as fast as smaller molecules. Western blotting, used to confirm the protein identity. Synthetic or animal-derived antibodies are created that react with a specific target protein that allows the visualization of the protein. Results. Figure 1. The composition of human milk is influenced by many factors: genetics, lactation period, dietary habits. These factors produce differences between one milk to another, including the antiretrovirus activity. Even though there were no clear differences between the 10 healthy donors in the electrophoretic profile of human milk fractions, there were significant differences in the antiretroviral activity. These similarities can be observed in the electrophoresis, where there aren't big differences between the bands. Figure 2. Taking into consideration these differences, the milk of the 10 donors were pooled. The electrophoretic profile showed that the majority of proteins present in the human milk are the whey proteins, band 3, especially immunoglobulin A, band 6, LF, band 7, Lys, band 8, and alpha LA, band 9. Caseins, band 4, and MFGM, band 5, that includes MUC band 10 and LDH band 11 represent lower proportion. As you can see, the higher in concentration, higher intensity, being alpha LA, immunoglobulin A, LF, Lys more intense, and casein and MFGM less intense. Figure 3. In order to guarantee the hygienic quality of human milk, a pasteurization process is applied in human milk banks. However, it has an inconvenience. The heat that is applied in the process may lead to a decrease of its beneficial and protective properties. They apply different temperatures during different times and evaluate the effect of heat treatment on the neutralizing activity of human whey against rotavirus. Heat treatments between 72 and 85 centigrades induce higher losses of activity, band 6 to 13. This is illustrated with the thickness of the bands in the electrophoresis. In 0, wherein applied any heat, so is the most thick, but 6, 7, 9 to 13 are the most thin, which indicates it has lost part of the neutralization activity. Figure 4. Immunoglobulin A and LF are the proteins that mainly show antiretrovirus activity. They applied heat and found out that in both proteins their protective property was reduced. This is shown in the electrophoresis by the decrease of intensity. Figure 5. There's another method to guarantee the hygienic quality of human milk called high hydrostatic pressure, HHP. The HHP treatment was applied to the milk in different pressures and time. Then they evaluated the antiretroviral activity and found out that this method was slightly harmful. In electrophoresis were changes of thickness in the electrophoretic bands. Figure 6. The effect of HHP on immunoglobulin A and LF was different. Seems to be more harmful to immunoglobulin A than to LF. This was illustrated in the electrophoresis where there was no change in intensity in LF but there was in immunoglobulin A. Discussion. Author Peterson, what he said, the majority of the human milk proteins are the whey proteins, the most abundant being alpha LA, LF, immunoglobulin A, and Lys. In this study, they obtained the same result. Superti. There is no correlation between the degree of LF iron saturation and its antiretroviral activity. In this study, they obtained the same result. Jolken attributed antiretroviral activity to the human milk mucin complex fraction containing the MFGM protein MUC1. In this study, they obtained the same result. Conclusion. Besides the fact that a human milk is fundamental for the right development of an infant, it's a natural way to protect him from the attack of pathogens because of the proteins it has. Because of that, it's important that the milk banks use HHP instead of heat treatment to ensure the protective properties it has. Of all milk fractions, the one that has most antiretroviral activity is the whey, especially immunoglobulin A and LF.